Right, so we have our large and small test tubes taken out of the water bath, and now in quick succession, add the contents of the small test tube, which is just the potato catalase, into its respective large test tube that it's next to. And you'll want to do this in quick succession so that we have as similar of a start time as possible between all 5 test tubes. So we're going to go ahead and add, remember A represents our cold, approximately 10 degrees temperature from the ice water. B is going to represent our room temperature, approximately 20 degrees. C will be from our 30-degree water bath, and D will be from our 40-degree water bath. And then finally, E is our warmest temperature on the range, and that will be from our 50-degree water bath. And after 4 minutes, we will have the opportunity to collect our data, which is our dependent variable. We're going to measure the height of the oxygen foam. All right, at this point, when you've reached the end of your 4 minutes, in quick succession, as fast as possible, you want to go ahead and measure from the bottom of the oxygen foam, the white foam, to the top. Remember not to measure from the bottom of the test tube. You're measuring from the bottom of the white foam to the top. So you'll measure for A and write down that measurement, remembering that A is our ice water, approximately 10 degrees, and then you'll measure B, and remembering that is from our room temperature environment, which is about 20 degrees Celsius, and then you'll measure C, and that is from our first water bath of 30 degrees Celsius, and then you'll measure D, and D is from our 40-degree water bath, and finally our warmest temperature, E, you'll measure that and make sure you record your data.